Hello.
Ooh. Hello, Benjamin. Can you hear me?
Yes, I can.
So I think everything works.
Okay. Hey, Mark.
Hello.
Hello.
How goes it?
Good. So, Mark is our sponsor for today's webinar. He'll have about 5 minutes to give a little introduction, and then we'll go with your presentation afterwards.
Okay.
So, Mauro, I do prevention programs for aquatic invasive species across the continent here. So I'm just going to give a little one-over on prevention actions as we think about the context of the golden mussels new introduction, and then talk a little bit about the technologies that are available, and then assess the greater picture and say, here's where we've done our work, and here are all the boat accesses within your region. And I'm going to pass this off to Mauro to talk about the true threat of this species in this context. Is how I'm going to do the transitional line, if that works for you.
Yes.
Great. Okay.
Cool.
Do you want to do the introduction as well? Just like a brief introduction for him? And then I'll jump in.
Sure, if you'd like me to. So I'll stick, hopefully I'll be under 5.
Okay.
Context. And then do you have something I can read for that?
Just, I guess, from the email, if you have a copy of the email or not.
Let me pull it up here. All right. So did you read the bio?
Yeah, sure. Or yeah, I think so. I mean, the bio is a little bit long.
Look, guys, we've been studying this for a long time. I think this is the main point. So I have a science background. I'm a biologist. I have a PhD. I teach at the university, and I also have a biotech company, which is the inside where we develop this solution, because otherwise it's very hard to raise the funds to do this kind of research. So if you go as simple as scientist and entrepreneur, it's good for me.
Okay.
Super.
Maybe one achievement that I think explains a lot is the fact that we sequence the genome of this species. So maybe this, for those who want authority, gives authority, and also explain that we are the genome is a important landmark for the study of a species, and so it says how serious we are about studying it, understanding it, and fighting it.
Okay, great.
Great. I can do that, Mauro.
Okay. Mark, are you going to be sharing your screen at all?
Yes.
Okay.
All I'm going to do is then take over.
Yes. Why don't you share your screen now?
Okay. So do you want me to keep it on or just keep the...
Yeah, just keep it on. We'll get started at 11.
Sounds great. All right, are we ready to get started here?
Yeah, let's give it another minute, maybe.
Okay.
Some people to fill in.
Well, welcome everybody. While we wait for people to fill in, I think you all are in a warmer place than I am. I'm Mark Apfelbacher. I'm with CD3 Systems and sponsoring this. It's 2 degrees in Minnesota right now. What is your guys' temperature? Anybody over freezing? Raise your hand. Show of hands here. I think most people.
We are like way over. We are very, very hot. Today is a very hot day in Rio.
In Rio, I suppose. Yeah. It's fantastically hot. And I hope I'm thinking high moisture and rain clouds for anyone that's in Southern California right now. I'm sorry to hear that there's more fires popping up down there. So I'm sending some cold weather that direction that I'm trying to do mentally.
All right, well, let's get started. Thank you all for joining us today. My name is Ben. I am the webinar host, and we have some very special people with us today. We're here to talk about Golden Mussels, and our sponsor today is CD3 with Mark. Mark, would you like to give us a little presentation on what you're all about?
Thanks, Ben. Happy to be here and happy to be a part of Calms, and thanks for all the work that you all do. I'll be really brief, just kind of an overview on what I do related to invasive species prevention. I'm based in Minnesota, as I mentioned, and I work on wireless cleaning systems as a prevention tool to contain the spread of new infestations. So I work across the entire continent, and the big thing that I work on is working on that message to help create a strategy, helping people plan to act, and when they don't act, we help them take those actions of cleaning, draining, and drying. CD3 Systems are waterless cleaning technologies installed at a boat launch. We're part of the AIS treatment hierarchy. As I like to say, we're not the silver bullet, but we're part of the silver buckshot. It takes a lot of strategies to change the behaviors of the boating public. So it's self-service equipment, it's waterless, it's 24/7 available, installed as infrastructure at a boat launch. And so with the equipment, they're solar powered, they're used by the public to take those actions. It's custom wrapped. We are on the front edge of a lot of outbreaks currently right now with the zebra mussels in Colorado, zebra mussels up in the Columbia. We've done some very large projects for containment of the spread of starry stonewort in Minnesota in the upper Midwest. And now we're working on some large projects with the hybrid hydrilla in the New England states. So the reason why it's wireless is so the public can use it. The hot water decon systems are great, but you have to have them staffed. There's an air blower system that's on the left-hand side. The air blower system is accompanied by push button operation. Got hand tools in the middle that'll tethered, and then a vacuum system that everybody knows how to use. So with a CD3 system, we've got different model types, different equipment specifications, but they all connect to the cloud. So you get data on when and how they're used. So as you think about your programs, know that you've got partners like us that are innovators in this technology space. I have the pleasure of working across the country with a lot of different cool projects. So we have a couple of units in the region in the New Melones area. These are all the CD3 Systems New Melones. Then we have some up in Tahoe. But as I transition here to the meat and potatoes of the presentation, I want to share one important slide here on the boat accesses in the region. So let me pull it up quick. So this is an interesting way to think about context. We're going to learn a lot about from Dr. Rebelo here on what these golden mussels are and the genome sequence and some of the challenges that have faced his region. But as we look into the context of, my Google Earth isn't working. I'll just stop presenting that slide. As we look into where those boat accesses are, let's think about prevention strategies because we don't want this to go elsewhere. So with that said, I'm going to transition the mic here to Dr. Rebelo. He's a fellow entrepreneur. He just told us that he's in Rio, so I'm glad you're in a warm place. His knowledge set includes sequencing the genome of golden mussels, and is going to share some stories on his technologies, his strategies, and approaches with that. So thank you so much for sharing us with your knowledge on this, and looking forward to the presentation. So take it away, Doctor.
Thank you, Mark. Thank you, Benjamin, for the invitation. I have big part of my team here watching this seminar. So like this, it's me presenting, but a lot of people that over the last 20 years contributed to the understanding that we today have of the golden mussel. So I'll touch into 6 points today, a little bit of the history of the invasion in Latin America. I put as a question if the mussel is a problem or not. You'll be able to see for yourselves from the pictures we are going to show. It's still a problem or not, it's still spreading. And this is the reason why we are developing the technology that we are, which is the gene drive to control the mussel infestation. I'll tell a little bit where we are now in this development, and briefly the challenges that we have ahead. I hope I'll be brief because I'm more interested in exchanging with you questions, and if there is anything we can collaborate to, I don't know if we'll be able to prevent further dispersion, unfortunately, but how we can think on medium to long term on how to fight these and other infestations. So for those who don't know, the mussel has arrived in South America in 1991. Here, sorry. In Buenos Aires, and it probably came with ballast water from China. So we know the mussel came from China in ballast water. As we know, it's original from China. And after many years of research, we were able to get in touch with researchers from China and learn that even in China, it's an invader. So it's original from the Pearl River basin and have spread over China through the Yellow River and the Yangtze, and it could not move further north because of geographical accidents that prevent its dispersal. So it started in Buenos Aires in 1991, but in 2001, it was already in the Paraná River in Brazil, in Itaipu, which is the largest hydroelectric power plant in the world. There's a second invasion here in Rio Grande at the Patos Lagoon. We know it's a second invasion that started in 1998. There's a big harbor here in Porto Alegre, and again, it came with ballast water. From Buenos Aires, it took two routes, one to the left in the Paraná River and one to the right in the Uruguay River, and it has spreaded upriver in these two very large basins. And at the time, nobody knew exactly how it was moving so fast upstream, right? Because you would expect the larvae to be brought downstream, and it was very curious because when you talk to people at Itaipu, what they tell you is that they were monitoring the river downstream the dam and they found first the mussel upstream already in the reservoir. And it was like nobody knew how that could happen, that they could not see it arriving, and when they saw it was already there. And what people learned afterwards is that it was moving basically through land with fishery, with recreational fishing boats, right? So people would put their canoes or their light boats into part of the river. The larvae would attach, the mussel would attach, then they would drive, I don't know, 200 kilometers upstream, go to the reservoir, and then those larvae would, that's how it was moving upstream. And what many years later, so today you have the mussel that's traveled like 3,000 kilometers, 5,000 kilometers, about 3,000 miles upstream, arriving to the northern part of, or to the central part of Brazil in the wetlands of Pantanal, but it has also moved towards the ocean, so infested big rivers like the Tietê River in São Paulo, and now in the northeast of Brazil, also the São Francisco River. So if you're curious, here around Cuiabá, you have presence of the mussel, and just 100 kilometers, or almost less than 200 kilometers, you have the first rivers of the Amazon basin. So like it's, the good thing is that it has been held in this area for the last at least 10 years. And one of the reasons why it's probably there, it's because since it's a wetland, you know, every year when the river comes down, it seems to kill a lot of mussels, and this is keeping the population under control in this northern limit. However, if you go to the right towards the ocean, then you don't have wetlands anymore, and you start to have a river like the São Francisco River that it's more permanent and got infested very fast. So the question whether the mussel is a problem, it's easy to answer when you look at images like this one. So this is in one of the hydroelectric reservoirs in São Paulo.
Showing the map.
Sorry. So this is a small sample of how the mussel can attach to everything. So this is when the reservoir, it was during the drought periods that the reservoir was down, and you can see that everything that is not natural substrate, they attach. They are a big problem for native mussels. And so this is one onion shell with a lot of golden mussel on its top. You guys probably seen it with the zebra mussel. It does the same. They have a very similar behavior. And these are fish that were fished in the Paraná River. So it's not true that the mussel doesn't have predators. So it is a very abundant resource, a food resource, and the fish food on it, but it's very hard to digest. So you can find fish like this one with the abdomen very enlarged because they ate a lot of mussels. When you open, the intestines are full of mussels, and there are videos of these mussels that were taken out of the digestive system of this fish and put in water, and they open and start to filter. So they were alive, and there is evidence that the fish going upriver and dying and are also a way of transporting the mussels upstream. So this is in a hydroelectric power plant reservoir in the Paraná River. This is a fish farm. They are raising tilapia here, and if you take this net tanks out of the water, they are completely clogged with golden mussel. The nets, they become so heavy that they can sink and let all the fish escape, or in other situations, they block the water renewal, and oxygen in the water can go down and kill all the fish. In this image in the right, you can see that we are looking at, there's almost no space in between the intervals of the net. They grow very small, and they come to the point that they completely clog the net. It's extremely hard to clean this. So what they do is they just leave for 4 or 5 days on the surface, like under the sun, and let them, die and fall back into the water. But it's a even bigger problem for the hydroelectric power plants, right? So this is an image of the intake of the grids that protect the intake of the water for the power plant. And you can see these are major structure. They weigh more than 2 tons, and you need a crane to raise them. And when you do, they are completely clogged with golden mussel. For those who want to see, there is a not so short video of the process of lifting the grid, and of the man cleaning manually cleaning the grid to remove tons and tons and tons of golden mussel. An extra challenge is how bad it smells, right? So it's a terrible smell, and it's almost an occupational hazard for them. But the bigger problem is here in the cooling systems of the turbines. So this green structure that you are seeing are the compartment in which the filter of raw water that is coming from the reservoir to cool the turbine. So the compartment and the filter they are cleaned every 15 days because they completely clog with the mussel, right? So you can see here in the left image, the filter just after it was removed from the compartment. Now the worker is manually removing, and then you have in the end after they removed. And below you have a video for those who want to check. This presentation is online, and you can check a video of the whole process of removing and cleaning the filter. So in a hydroelectric power plant like this one, they monitor this the risk of overheating because of the clogging of the filters. So every 15 days they are doing maintenance. But there are cases that people didn't expect. They were not paying attention to the mussel, and they just discovered when the turbines overheated, and they had to bring down the whole power plant for risk of overheating. So it is a big risk. Other tubes, other pipes are also clogged with the golden mussel. But again, a situation very similar to the one that in North America you see with the zebra mussel. This was a very impressive image for me. Even it's not a very good image, because what you're seeing is the door that closes the compartment of water after the turbine. And from time to time, they have to give maintenance to the turbine, and they need to close this door. And this is a gigantic gate, and they could not close the gate because of the golden mussel that infested the whole area of the tracks where you would bring the gate down. So, like I said, it's the current technology cannot stop it, could not stop it for the last 20 years. When we started work with the golden mussel in 25, we were specialists in molecular biology of bivalves. We were happy working with oysters, and we didn't really. We were not paying attention to the mussel, even though it was around for more than 10 years. But then we were invited, because we knew how to do molecular biology of bivalves, we were invited to participate in a task force to try to develop control solutions. But most people were looking at poisons and how to dilute different substances and how you could come up with either chlorine strategy or different compounds that could kill the mussel. And it doesn't seem to, whatever it is, it can work in the lab, but when you bring it to the field, it's either very difficult to deploy or it's very expensive. And we even tested microencapsulated compounds, biobullets, and even though they may work inside the pipes and in specific situations, it's like wiping ice. At least that's how it feels for everyone involved. So it dispersed to the San Francisco River. It arrived in Rio de Janeiro. There's which is a completely different river basin with like mountains in. So the river basins are not connected naturally, and now it's even in California. So what we saw is that if before recreational fisheries were the main vectors to transport the mussel, now it's aquaculture. So we know that two things are happening in aquaculture. One, the alevins that are sold to farms in different states are bringing the larvae in the water. So there's more than one evidence of alevins transporting the mussel together, and there's evidence that this is how it arrived in Rio de Janeiro state. But on the other hand, but also on the top of it, the aquaculture structures are a vector in itself. So these two images are showing monitoring of the distribution of golden mussel in a reservoir and in a in a river basin. Okay, so this let's say this was a way to quantify the infestation using video, sound, And at the time, we didn't use eDNA that we are also using now, eDNA, but it was a method to quantify the infestation using both sonar and image. And what we saw was that wherever you had artificial structures, you had a higher density of the mussel, right? So what we are seeing is that the mussel is a very low signal in these huge water bodies. It's constant, but it's very low signal. And if you put an artificial structure on it, it's going to amplify the signal, and these areas are going to become the nurseries of the mussels that are going to infest. So if you like, it can jump from one area, one artificial substrate to the other. This is the main way of dispersing inside the river basin. So we were not happy with the strategies that were being, it looked like people just wanted, in the lack of a better way to do things, they were simply doing more of the same, which is, let's try to find a new substance that can better kill or kill more or is cheaper. And instead, we thought, okay, maybe because of our bias towards biotechnology, we said, okay, let's give a step back. Let's assume that poison is not going to work in the water. What could be a different approach to solve this problem, from first principles? And we started seeing like we don't know anything about this species, at least compared to the knowledge that is available for traditional biomedical models. There was very, very little information about the mussel. So we started by sequencing its genome. So we started with the transcriptome, and then we moved it to the genome. For those that are curious, there is a very cute video that tells the story of the genome, and it was the video that we used in a crowdfunding campaign. The first version of the Golden Mussel genome was crowdfunded, and we published it in 2017. And it was a draft genome, and we soon started to realize it was not good enough to, as I'll tell, it was not good enough to find the target genes that we wanted to use in the gene drive. So soon after publishing the first genome, we started a high-quality platinum genome that was published in 2024. But when we started the genome, we still didn't have a model of how we would control the mussel. Our first idea was environmental RNA interference that has a lot of challenges, but still is the strategy that the group in University of Minnesota is pursuing for the zebra mussel. So, and they have at the time we were saying, okay, how we are going to encapsulate RNA, and that is going to arrive to inside the mussel that is inside a pipe in a power plant. And they are actually using bacteria inside algae that will be the vectors for the RNA. So very good ideas around in strategies to try, in biotech strategies to control invasive bivalves. So you have this environmental DNA. There's another group that is developing, there's pursuing virus approach. They're trying to use viruses that induce cancer or a kind of cancer in clams, to modify these viruses to control zebra mussel. We, on the other hand, read the Anopheles gene drive paper that was published in 2016, and I said, wow, okay, this is something that we could adapt for the golden mussel. I was in an event that the researchers that published the Anopheles gene drive were, and I started to talk to them, and they said, look, this is a good idea, let's do this. And since then, we started to see how we could adapt the same model of gene drive that was being developed by the group in the Imperial College in London to the golden mussel. In 2017, then we start an R&D program within the framework of the ANEEL, is the regulatory agency for electric energy in Brazil. So it's a kind of your DOE, and they have a program for the hydroelectric power plants to invest in R&D. And then we started the project with 3 hydroelectric power plants that invested about $2 million over the last 7 years in the in developing this idea, which is, let's find a gene that will block reproduction, the ability of the mussel to reproduce. Of course, you cannot just make the mussel infertile, otherwise you don't spread the trait in the population. So I will explain a little bit better, try to explain very briefly how a gene drive works. I'm sorry that this infographic is in Portuguese, but I'll walk you through the process. So the first step is to genetically modify the sperm or the egg of the mussel with a gene that is to make that will make an organism that is non-homozygote for the trait, right? So how does Mendelian inheritance works? If you have a mutation here represented in a blue mussel, when you mate the 2 mussels, this mutation tends to be divided by half in the offspring, right? And then when you bring other wild mussels, over time, this mutation tends to dilute until it disappears in the population. Of course, if it doesn't bring any gain in fitness. With the gene drive, on the other hand, you create an heterozygote organism, and this modification actually will correct the other allele, or will transform the other allele of the gene. So in the second generation, it's, instead of being diluted, in the first generation, instead of being diluted by 50%, you have 100% of the offspring carrying the genetic modification. So when in a second generation you bring other organisms, wild organisms, to reproduce, again you have 100% of the offspring carrying the mutation, until you get to a point in which the heterozygote is so abundant in the population that they start to reproduce with each other, to mate with each other. And when you have 2 organisms carrying the genetic modification, the resulting offspring is infertile. So the trend is that over time, you are going to collapse the population simply by blocking the reproduction. So it's important that It's not the organism that we are creating is not infertile in itself, but it carries the gene that when an organism is homozygote for that trait, then it becomes infertile. Where we are now, so we have chosen the genes that are going to, that we will modify. So the gene is called DMRT1. It's a dimorphic, it's a double sex dimorphism gene. It's basically the same gene that was used in the Anopheles mosquito. And we have built the constructs for the gene drive. We succeeded in transforming the embryos with this construct, but right now we haven't reached homologous recombination, which means we still don't have the gene carrying a genetically modified mussel that is carrying the CRISPR construct. But we have succeeded in many ways in transforming mussels to show that we have all the right molecular tools to do the modifications. So now it's about, because homologous recombination has a very low success rate, we have to do a lot of transformations to succeed in generating what is going to be our matrix, right? Our matrix organisms. And this was, a big challenge to achieve this, to build this prototype. It's actually to reproduce invasive species in captivity in the lab, right? I know that it's a similar problem for groups working with zebra mussel. Regardless of the extreme success of these organisms in the wild, when you bring them to the lab, they don't reproduce. Very hard to reproduce. So we have worked very hard to reproduce these organisms in the lab. So what you're seeing is an image of a mussel that is 4 years old. We managed to 4 months old. They survived, I think, this one until 7 or 9 months. And we managed to, if you see the graph here on your right, we managed to scale up the production of embryos. So now we are in the position to have enough embryos to modify them at scale in the lab and go for the homologous recombination of the CRISPR construct. And this is a very preliminary modeling of the dispersion of the gene drive in the population. So imagining that we will eventually succeed with the transformation and the creation of the organism, how it would spread in the population. And this model is very limited. It's based on assumptions of mosquitoes, and it has several other assumptions that we had to make, not necessarily based on evidence from the mussels, but it gave us a tool that we can improve over time to make estimations about the dispersion that we are going to validate first in the lab and then in the field. So with this model, this is not, so you're seeing like an image in one of the block, how it would spread over time. And with the assumptions that we used, eventually it would disperse, spread, and dominate the population. But it could take a long time. Like if we start from a reservoir with high biomass of mussels, it could take up to 10 years to completely to spread and control the population. On the other hand, if we could somehow reduce the population of mussels and then apply the gene drive, the model shows that it could be controlled in 2 years. So even though it's hard to imagine how we could, it's hard to imagine how artificially we could control or like massively kill mussel in a large reservoir. It's true that if you have a natural event like a drought, that these natural events, they kill a lot of the resident mussels in a reservoir. So it could be the case that we would wait for a natural event that would reduce the water volume of the reservoir, eliminate a lot of the mussels, and then we would apply the gene drive as a way to speed up the control. This is how we are imagining to control a reservoir that has been invaded. But maybe the best use of this technology would be to prevent a basin that hasn't been infested, or a reservoir that hasn't, a river that hasn't been infested, from becoming infested. So you can imagine some of the ideas that we are exploring is like cultivating genetically modified golden mussel in rivers that cannot reproduce by themselves in these reservoirs, so they would work as a wall, right, to block any mussels coming from like any eggs that are coming with the water. So a lot of ideas on how it would be applied in the environment. And to finish, the challenges that we face ahead, like I said, this is an R&D program of the energy sector, and this is not a sector that is biotech friendly. So when you come to them and you say, look, I need like $10 million to further develop these are, even though, I don't know, for pharma industry or for agriculture industry, so sectors that are familiar with the costs of developing a genetically modified organism, going through all the safety and efficacy and regulatory steps for it to be safe to be released in the environment, they are more familiar with this, with the investment required to do it. But for the energy sector, it has been a challenge to accept that they have to make investments of this order of magnitude. The second thing is that they see themselves as a victim, which in many ways they are, of the mussel, and they don't see why they should be held responsible for developing a solution that will benefit not only their sector, but everyone. But you know, these are a bit political discussions, but the fact is that despite the $2 million that have been invested so far, we still need a lot of money to further develop the technology. So if we manage to raise this money, continue raising the money, the big technological challenge now is the homologous, in a way, the rich homologous recombination, which will allow us to go to the next step, which is to do all the efficacy and safety tests that are required to submit a request to deploy the mussel in the field. We know Brazil has a very progressive regulatory environment regarding genetically modified organisms, and we believe that we have the example of the genetically modified mosquitoes for dengue fever. That's a completely different technology from Oxitec, but that was first approved in Brazil. We have now, genetically modified fish. We have, not to mention all the crops and the livestock. So we believe that if there is a place where this can be accomplished, first, this is Brazil, and this gives us some competitive advantage over groups that may want to develop this somewhere else. But in the end, a big question is, if we develop this, if it's safe, if it works, who is going to buy it? Who will be responsible for paying for the deployment of this in the field? There is a lot of discussion, like I said, in the electric sector, a lot of evidence that regulatory agencies somehow will push this to the electricity sector to pay this bill, but this is not clear yet. And this is one of the reasons why it's also still hard to raise money, right? Because no one knows who is going to be the client in the end. I think I spoke maybe a little too much, so I will leave now. I'll be happy to answer any questions that you have.
Okay. Mauro, thank you so much. I have to say that I made a really big mistake, and the presentation was announced to start at 12 noon. So even though it is 11, or it's said it was supposed to start at 11, everyone's starting to show up right now. So Mauro, I'm terribly sorry. Would you be able to give this presentation again?
Some other time, not today.
Okay. All right. Well, luckily we have this recorded, and so we'll share the recording. So I guess let's start the discussion, and then when you leave, we can just, or perhaps someone on your team can kind of give a recap of what we missed out.
Well, so I can go until 5:15, so like maybe a little bit less than half an hour. But Daniele, Juliana, Yasmin, Fabio, they know even more than me about this, so they are able to answer any question, especially if are technical and scientific ones.
Okay. All right. Again, terribly sorry. I take full responsibility. I was so confused as to why nobody was coming, and then I looked at the announcement and I was like, oh shoot, we got the announcement wrong. So that is my bad. So I'm sorry. But okay, well, I've got a few questions since we're here in the discussion. So one of the things you mentioned was that there were fish that eat the mussels. Do these fish normally eat bivalves or similar things, or are they just generalists?
They're generalists.
Okay. Because we have fish here in California, but really not many that are adapted to eat the mussels. So I wonder whether or not that would be a method or a vector of transport or spread in California.
These fish were not adapted. Also, they just see the resource and they prey on it, and that's why they are dying of preying on it. Mm-hmm. So this can be, I believe you may find the similar problem. because it will be it will impact the fish populations, and it will also be a vector.
Okay. Okay. All right. I've got one more question. So, as I understand it, there's a virus that you deploy that basically modifies the gene in the population. How is the virus deployed?
No, this is not our approach. There is a group in US that is developing this approach of the virus for zebra mussel. What we do is transformation. Now we are using electroporation so you synthesize the gene construct synthetically, and you take the DNA and transform using electroporation inside the embryos, or the embryos, or the sperm, or the eggs, right? And this was part of the big challenge, not to fry the eggs or the embryos with the electroporation, and the rates of success are very, very low, and that's why scaling up and reproducing the mussel is so important if you want to develop any biotech approach solution.
Okay, thank you. We've got some questions. The first question that came up is from Tracy. Is there proof that a decontamination of a certain temperature can kill veligers or adult mussels?
I'm not that I'm aware of. So we know, I mean, probably in the lab, but in an industrial facility, we know of studies that try to control larvae with temperature and with ultrasound and with UV, and they may work in one trial or another, but I'm not aware of successful attempts to control in a power plant with temperature.
Okay. Lee, I see you've got a hand raised. Do you have a question?
Yeah, I've read in publications from Fish and Game that these mussels can't reproduce or spread in water temps below 60.8 degrees Fahrenheit. And I was wondering if that's true.
So sorry, how 68 is around?
That is 15.5 degrees Celsius.
15. Danny, do you know? I think they do in Argentina. In Argentina, the temperature
it's lower than here. Yeah.
Yeah.
They are very generalist. They tolerate salinity and a broad range of temperature.
So in Argentina, they get the mussels from the wild at 16 degrees Celsius, and they induce spawning by raising the temperature to 19. So I think they would reproduce around 15.
I don't know. Just according to publications for the California Department of Fish and Wildlife, it basically pinned them at 60.8 and below. They couldn't reproduce or spread at that point. So that was just a big curiosity of mine, because there's this huge worry right now, and all the lakes are closing down, but most everybody's water temp, surface temp, even the rivers are at about 50 to 54 degrees Fahrenheit. I'm not sure what that's Celsius, but I'm just trying to find out where does the risk really rise exponentially? At what point, what temperature do we really have a big worry on our hands?
So I'm this website with so there's a group in Brazil that developed a risk index model, and it's this page that I'm sharing with you that showed pinpointed all the points that have infestation in Brazil. So maybe you can play with this model to see if it can help, like accessing infestation risk or dispersion risk. What we see, our situation is very particular. It's hot all year long, and what we see different from Argentina, they reproduce here all year long. So they reproduce multiple times a year. You may find a peak during summer, and it may go down during the winter, but they can reproduce all year long. And an interesting fact that we still don't have like abundant evidence, but we have enough evidence to further explore, is that they may reverse sex with temperature or food abundance, like oysters do. So there are, if you go to a mussel bank during one period of the year and you come back to that same mussel bank in another period, the male-female rates can completely reverse, like 2-to-1, 3-to-1, and then 1-to-3. And we still don't know how these impact in dispersal, but it's something that it has been observed, like changing in the male-female rates at the same mussel banks over the year, throughout the year.
Okay, we have another question from Tracy. Do we know how long a mussel can live out of the water?
We never tested this exactly, but I know anecdotal evidence for zebra mussel of them living 2 weeks in the pocket of a researcher. It's of course, when you want to bring them from where they are to your lab, you may have massive mortalities. But if you just, like forget one somewhere, it may go for a long time. So I'll try to answer this. In the water, we know that it can close the valves and stay with the valves closed for up to 30 days. And this we know because that's how, when you put, when you try to control them with poison, so there's more than one study showing that they can keep their valves closed for a long, long time. And this is assumed to be one of the reasons why it's so difficult to control them with chemicals in the water. As soon as they see chlorine, they sense chlorine, they close the valves, and they can stay with the valves closed until you stop putting the chlorine, and then they open up again and they go.
Okay, for everyone joining us, we have an announcement to make. So the presentation has been recorded, and the initial explanation has already been given. Unfortunately, Mauro is running out of time, so I'm kindly asking that he just talk a little bit about the gene drive, and then after he leaves, we can talk a little bit about what is happening in Brazil. I think there's some other colleagues here with him that might be able to help out and give a little bit of a explanation as to what else that Mauro can't cover. So my apologies for this mistake, but let me introduce our sponsor and our presenter. Today we have Mark Apfelbacher with CD3, and we will give a little video or give him some time afterwards to give a talk. But let's just go straight to the presentation. Mauro is a scientist and entrepreneur. He is been studying the golden mussels for many, many years in Brazil, and he's got a really innovative take regarding the genetic gene drive for golden mussels. So Mauro, please take it away.
Oh, okay. I'll show two pictures. I will assume. So unfortunately, we had this problem with the time, and mussels have arrived in Latin America, in Buenos Aires, in 1991, in ballast water coming from China. Since then, in the last 35 years, it moved 3,000 miles upstream up to the wetlands of Pantanal in the border of the Amazon ecosystem. It has also moved east, following different river basins, and it has invaded most of the river basins in southern Brazil, and now it's also in the northeast states. So the main reason for this is, one, it's hot in Brazil all year long. They differently from zebra mussel, they reproduce all year long in our temperatures. They attach to ship hulls, to different boat hulls, and then if you move the boats through land, it's a main vector of dispersion. And now we also know that aquaculture alevins that are transported from invaded areas to pristine areas are a main vector of invasion. And over this period of time, many people have tried to apply the, let's say, traditional strategy for controlling invasive species inland, which is to use poison. And in the water, it's much harder. It simply doesn't work, right? So the feeling that we had when we first started to work with the mussel is that we were wiping ice, and this was not going to work, which motivated us to go back to first principles. How can we imagine a biotech strategy that would once and for all be able to control the mussel? And to do that, we sequenced the genome in a way to get a lot of understanding of this organism in the shortest possible period of time, and to find targets that could be somehow used to control the mussel. But we still lacked a framework strategy that we could deploy all this knowledge that we were generating. And this came in 2016 when the Anopheles gene drive mosquito was published, and we got in contact with the same group, and we started to work ourselves in a gene drive for the golden mussel. So usually inheritance grows through Mendelian way, which means that if you have a mutation, here in this image represented by the blue mussel, it gets diluted in the next generation because only 50% of the offspring is going to carry the mutation, right? So when you bring new wild mussels to this second generation, the mutation will be further diluted. Of course, if you don't have any gain in fitness, the trend is that the mutation will be diluted and eventually will disappear. With the gene drive, on the other hand, we have a mutation that copies itself, actually transforms the allele into, it transforms the allele, copying the modification that we induced. So over time, instead of a dilution of the trait, you have a concentration of the trait. We call this super Mendelian inheritance or gene drive. So what we did was to choose one gene that Juliana and Danny can tell more about it after I leave, which is the DMRT1 gene, and we are modifying sperm and eggs with this gene or embryos, and in this way creating a muscle that is heterozygotic for this trait, which is a gene that blocks reproduction. So the idea is the muscle itself is able to reproduce with a wild one. That's what we are seeing in the top of this figure. But then the offspring, instead of being 50% wild, 50% modified, they are 100% modified, because the gene drive is going to act to transform the allele, so the muscle becomes homozygotic for this trait. Then the second generation is going to mate with other wild muscles, and then again, the third offspring will be 100% heterozygotic. You can bring more muscles, and you go on, and this way you increase the frequency of the genetic modification in the population, and eventually these muscles become dominant and they start to mate among themselves. And when this happens, then you have a muscle that is born homozygotic for the gene that blocks reproduction, and this muscle is sterile. And the idea is that through through some generations, you reach a point where the population collapses for lack of reproduction. So this is a bold idea. As far as we know, it hasn't been tested in the field anywhere. The Anopheles mosquito is very advanced in terms of efficacy and safety tests in the lab, but it faces major regulatory hurdles and bottlenecks to deploy it on the field. Like I was saying, Brazil has a very progressive regulatory framework when it comes to GMOs. So we believe we have a competitive advantage to be the first group to deploy a safe and efficacious gene drive to be tested in the wild, in the field. And we hope to do that. And the major challenges that we are facing now is to continue to raise funds to move the research forward, now that we are in the step of having modified genes, having modified muscles in the lab with GFP and with CRISPR constructs, but still not at scale.
Okay, great. I know I want to be very respectful of Mauro's time. We'll open it up for discussion right now. Unfortunately, there was a mistake in the announcement. We started about an hour early, and so for those of you joining us, we have a recorded session that we will send out. But again, so Mauro doesn't have much time left. Unfortunately, he has to leave. So if there are any questions, please ask now. Okay, Melinda, go ahead. See your hand is raised.
Thank you. And sorry, I actually did miss the talk. I just came on, so this might have been covered. But I'm wondering, for gene drive, something that I've heard is an impediment to it is the idea that if you introduce it into the wild, that somehow those organisms will get back to their native landscape and therefore could have the risk of causing species extinction. Is this a fear of yours, or is there something that could be done to prevent that possibility?
So there are a lot of theoretical things that can happen and a lot of theoretical solutions to these possible problems. One is called daisy chain gene drive, in which you would need, like, you have different triggers that make the gene drive less efficient, and you would only be able to reach a given kind of efficiency if you continuously deploy the gene drive in a certain area, and that would reduce to, it's already really unlikely that something like this could happen, but with the daisy chain or different triggers, you could reduce even more the risk of anything like this. If you talk to the gene drive community, the main fear that we have is that it's not going to work, meaning even if we succeed in creating the organism, it's not going to control the population. And the rest of the discussion, I believe, it is always influenced by the tolerance to risk that people have, right? And the tolerance to sacrifice, right? So if you have a big mussel problem, you may be more tolerant to risk than if you don't. But it's not a question that can be answered only by science, because even when science says, look, the probabilities are this and that, then people may choose an extremely conservative approach. What we see, for example, what I've presenced several discussions regarding the gene drive deployment for malaria in Africa, and it's a situation like this. Even if they have very like a debilitating country, like continent debilitating problem, there's still a lot of intolerance to risk to deploy this solution. This is why it hasn't been tested in the field yet. On the other hand, Brazil, with a, let's say, not as big problem for dengue fever, has allowed the testing of the self-limiting Aedes mosquito. It's true, it's not gene drive, it's let's say more complex technology than a self-limiting organism, but when you see the results, the self-limiting mosquito reducing the wild population, the urban populations of mosquitoes by 98%, and people simply not getting sick. I try to give a statement that is not biased, but I believe this is a technology that is worth being tested.
Okay. All right. Well, thanks again, Mauro. Is there anyone from Bio Bureau that can stick around for a discussion?
Hey guys, Juliana and Danny are going to take over.
Okay.
Now. I'm
All
very
right.
sorry I have to go, but I'd be happy to come back and present again and take more questions
Okay.
some other
And
time.
then if you can share an email, if anyone would like to email you questions, could you share that with us?
Yes. I'll put here. I see a question about the ballast water, for example, that I can answer by email. Thank you. Bye-bye.
All right. Thank you so much. So let's take a 1-minute break. Mark with CD3 is our sponsor for today. Mark, if you're listening, can you please unmute yourself and introduce yourself?
Hey everyone, I'm Mark Apfelbacher with CD3 Systems. Thanks again for the opportunity to support this important talk. Sorry again about the timing. And I was just going to give a little bit of overview on some of the work that I've done across the continent on prevention programs and initiatives. The big piece is that, I know you all are really concerned about the spread of aquatic invasive species throughout the region, but golden mussels present a new risk. And part of the challenge that you all face in California is that you're telling the public to clean, drain, dry, and dispose. And the big piece and what CALMS does is really help create partnerships like this to talk about prevention and management ideas. But when the public plans to act and you tell them to at a boat launch, they don't act. And there's been tons of research across the continent on behavior change, public boater awareness, and campaigns. And so C3 Systems, we are part of the treatment hierarchy. We aren't the silver bullet. We're part of the silver buckshot. We educate and empower the public by providing self-service cleaning equipment and digital resources for boaters to understand the best management practices that clean, drain, dry. It's infrastructure. It's a long-term effective tool at a boat launch that has 24/7 availability. We get data on the tool usage and efficacy related to that tool usage per watercraft, and it has 10+ year lifespan. So what it is infrastructure. We install these at infested water bodies. I myself am based in Minnesota, so there's two paradigms across the entire continent we work on. It's lake shielding and lake containment. Lake containment is containing the spread at an infested water body, thereby preventing the spread outward into a non-infested water body. I think shielding is the opposite. Are you shielding inbound watercraft versus outbound watercraft? Shielding, you're targeting the inbound watercraft to make sure that they are going out, getting cleaned on the way out. So with C3 Systems, we're waterless is the big piece of the puzzle, and how they work is that they have, if you walk up to a unit, it's an educational kiosk, custom wrapped with your call to action, QR codes. On the left-hand side, we have push button operation with an air blower system. This is 90 PSI air. Again, we're targeting all aquatic invasive species. We use this to target day boaters, people that are on a water body for less than 24 hours. Research across the continent has showed that if a boater is on a water body for less than 3 days, they can't have zebra mussels and quagga mussels in particular begin to harden on their watercraft. And where those aquatic invasive species will be is on mud, debris, and plants primarily. So cleaning, draining, and drying is a prerequisite for the 100th Meridian protocols for boaters to take actions on. And so we provide those tools with this equipment. So an air blower tool is push button operated. It automatically comes on and goes off. We have lights that illuminate into and under a watercraft with lights on the top and lights on the bottom. It's push button operated, automatically goes off too. And then we have a series of hand tools. Those hand tools are a brush, grabber, and plug wrench tool. Again, getting people to take those actions of clean, drain, dry. And then the right hand, and those tools are tethered with a braided stainless steel in the middle. They ratchet in, ratchet out. And then we have tools on the right-hand side that are the wet dry vacuum system. So the wet dry vacuum system is used to suck out any residual water, any residual mud, debris that they have in their watercraft. So they are completely dry and they don't have any vellagers in the system. There's different model types for different applications. And again, with your new infestation, you've got a lot of small accesses and you've got really big ones. The good news is we cross the spectrum of that. So with the equipment, you may ask, well, how do we know if it's working or they're cloud connected? C3 Systems provide a data access portal where you can understand tool use, tool counts, and when and how it's used during the course of a week. So with this talk, I was hoping to sponsor it because, one, I believe in columns. I believe in the community that you all can take the actions. And CA3 systems are across the country. We are in 33 states, 15 tribal nations, and 4 provinces. We've got CA3 systems up at New Melones Lake and Tahoe are the nearest to these infestations. And we hope to have a couple at the marinas that are nearby within the actual water body around Stockton. So with that, I'll transfer it back to Ben, but I'm really excited to support this and really want to help address you with the tools that can provide the actions of clean, drain, dry, and support prevention activities across the state. So Ben, you're going to send everybody the recorded presentation that goes after this, right?
Yes, and thanks again, Mark. You do have a question in the text chat, so feel free to respond to that. So let's, as it's already been discussed once already, and the recording will be sent out. But I think for those that are joining us, let's talk a little bit about how they are spreading. How are they moving upriver? And then we can talk a little bit about the challenges of control. And then, if there's any questions, feel free to just ask, and we can have more of a discussion. So Danielle or Juliana, can you tell us a little bit about how they're spreading in Brazil?
Yes. Do you have the presentation, Mauro's presentation?
Let me try to find, because there was a map there, right? Useful for that. Just a second.
Yes, but just a little bit of the story that Mauro told us. And the mussel arrived in Brazil in the 80s and came from China in ballast water, most probably. And it migrates from Argentina to Brazil. It's spreading up the river from the south of Brazil in direction to Amazon. So they are attached most in artificial substrates like material, like the structure of hydroelectric power plants in Brazil. So they are a big problem for hydroelectric power plants. And they cause clog in the system, in the water system, and there's a lot of time consuming and money consuming that they have to stop to cleaning the hydroelectric infrastructure. And that's a problem here in Brazil, as I mentioned, since the 80s, and it keeps spreading. Now we can find in the northeast of Brazil that they were probably transferred from the south to the north because of a program that hydroelectric power plants had to do. They have to repopulate the reservoirs with fingerlings. So the fingerlings are cultivated south, and then when they moved them to the north, they transported the golden mussel larvae too. So now it's spreading in the air. It's kind of jumping from rivers to rivers and spreading faster. And that's a short story.
Okay, no problem. So we're assuming that they're moving up with boats and contaminated water. One of the interesting things was that the fish were eating the mussels, and these were generalists, catfish that don't normally eat the mussels, but they somehow found it as a food source. Are fish transporting the mussels upstream?
Yes. Well, one of the problems, I don't think it's not the fish itself. Most of the times the water that's transported with the fish, but the animals that, for example, eat the fish, as Mauro showed before, if they die upstream the river, yes, they can spread the mussels there. The mussels are very resistant. They can survive inside the fish for a while. but are not the main vector for sure.
Okay. The main vector being what again?
Well, actually, boats are one of the vectors. And this transportation by human, as I mentioned, also, and mainly actually human things like, as I mentioned, the boats and tanks that are used to cultivate fish for feeding, for food, also are a problem.
Okay. All right. I saw someone had raised their hand. Is there a question out there? No? Okay. All right. Let's talk a little bit about the control. So I know Brazil has tried a lot of different control methods, some of which are kind of new to me. Can you talk a little bit about what's been tried before and how those have worked?
Well, here I think the main method have been used is chlorine, but there are also paints and manual removal, and it's basically physical chemicals methods. And there is nothing like we are developing or any other kind of biological control. So basically, I think it's very similar for what you have in US for the zebra mussel.
Mm-hmm.
Nothing new.
Okay. One of the things that I know has been tried out here for zebras and quaggas is the use of copper molluscicides. So they basically put copper into the water in hopes of killing the veligers and the adults. Is that something that has been tried in Brazil?
Do you know, Danny? I'm not aware of copper being used.
Yeah, I don't know. Maybe Fabio's still there. I don't know if Fabio's still there. But these different kind of things, it solves the problem for maybe a few days or a few months. So they come back again, maybe stronger than before, because when you use this kind of control, you also kill different things. And the problems, they reproduce very fast and they infest an area very fast. So these kind of things that kill the villagers, they will be able to reproduce again in a few months. Thousands of mussels.
Okay, thank you. Sierra asked, how long can the villagers live inside ballast in boats? Would a 30-day quarantine be long enough for them to die?
30 days? I don't think so. And we've been seeing that for more or less 30 to 60 days, they can keep the valves closed and they store the water, they stay alive. And then when they are in the friendly environment again, they start feeding and growing again. So 30 days, I don't think will be enough.
Okay.
Is it enough for the quagga mussel or for the zebra mussel?
That I don't know, but what I do know is, boats are supposed to discharge their ballast within, I think, or at least 100 nautical miles from shore, and correct me if I'm wrong, but the boats are supposed to discharge their ballast far enough away so they don't invade our freshwater systems. Okay. All right, someone says the 30-day quarantine would be dependent on the environmental conditions and time of year. Okay. Let's talk.
I'm Yasmin. I'm a colleague from Dani and Ju.
The person who was asking about the villagers, like we do not know for sure, like how many days the quarantine, how long it would take. What we used to do in the lab, once like we used the water from the tanks that we keep the mussels, we usually keep them in chlorine for about a week, or we can also use ozone. It also helps usually to decontaminate the water, but we never tested like how long they can be exposed to drought and still be alive, for example. I'm not sure. But yeah, and also like I said, they came from China to Argentina. I don't know like how long that trip usually takes, but usually it should be about a few weeks, and they were able to actually survive in half seawater and half fresh water. So they are pretty resistant to drought and salt and water.
Okay, Hans, go ahead.
Yes, I know this depends on conditions, but sort of in general, you said that they can colonize pretty fast. So what does that mean? So like within 3 months, you got a big population in your lake, or what?
Well, that could depend a lot. Most of the places where the mussel has been reported, like at first, the mussel started being reported in Argentina in 1991. So like we don't have track of how long it would take for him to, like, once he gets to the place, how long does it take for him to be like considerable to like affect the function of the place or of the power plant. But like we have, I'm not sure, like it depends a lot on the conditions of the water. The mussel usually likes to grow where there's a lot of organic matter available, usually that can help. Their life cycle is pretty quick. They could reach adulthood in 3 months. So basically, I would say 3 months would be like a comfortable setting period. I would say, I'm guessing, but we also like don't have that information for certain because it varies way too much on temperature, all of that.
Okay, thanks.
Okay. If there aren't any questions, I have a question. Okay, Patrick, go ahead.
Yes, can you put us in touch? Can you put us in touch with some of the hydroelectric facilities and maybe irrigators that are combating this mussel? Or could
Well,
you name a couple?
yeah, you can say it better than me.
Yes. We actually, our projects have been financed by 3 of these companies, so we can try to put you in contact if you want with one of them.
Okay.
You can send us an email so we can.
Yes, there's an email in the chat, so please go ahead and send that.
Mauro at the Bio Bureau. Is that the correct email to go through?
Yes.
Okay, thank you.
Okay. I'm a little curious to basically the status or the stage of your technology. So you've kind of described how the gene drive works. What stage of technology are you in? Like, is this ready for field trials, or where exactly are you with this?
Well, unfortunately, like we're not ready for field trials. As Mauro, I don't know if you got to see it, but the last stage where we got to was where we could find an embryo that was able to express, even for a short while, the GSP gene. So we saw like a transient expression. So we do not know for sure if the repair, the homologous repair actually happens. So we still need to optimize all of those stages, to be sure that we actually have been able to actually introduce the construct inside of the genome. But we're not like, we were at a lab prototype at best, but only at larvae, not ever on an adult individual.
Okay. Thank you. Mike McGraw, you have a question?
Yeah, the question I have is we've utilized the services of muscle dogs, in the company that we've utilized, and I know most of the companies are they've been certified where they're able to detect both the adults and the veliger, the larval form of quagga and zebra mussels. And right now, the vendor or contractor we've used, and I'm the park manager at a U.S. Bureau of Reclamation reservoir, New Maloney's here in California, and our vendor was provided with some adult golden mussels, and they were able to train their dogs, and the dogs are now certified to detect the adult golden mussels. However, they haven't been provided with any veligers yet. I don't know yet. In the probably when the water temperature increases in the delta, maybe that would be the mussels will start spawning. The veligers will become available, but as soon as our vendor is provided with those veligers, she can get her dogs trained. Now, this is a question for anybody from California Fish and Wildlife, as well as our folks from South America. But is there any way in the near term, in the near future, we'll be able to get some of those veliger, golden mussel veligers, so the dogs can be trained to hit on them, to be able to detect the larval form as well as the adult?
Well, I'm not sure. Like, I don't have any training in. I don't know anything about like dog training. But regarding like obtaining those veligers, we used to do the same thing here. Like, for example, our lab is in Rio de Janeiro, but we still do not have like mussel colonies here. So our mussels would come from the south, and we would send them like a bulk of them on to us, and then we would have to acclimate them, and we went to a lot of trouble to realize like what were the best conditions that we could keep them. And in the end, like it took us a real long time to actually know how to manage, like to do the thermic shock for them to actually be stressed, using also like chemicals to induce the spawning in the mussels. We now have a more comfortable protocol, but we based that on one from Argentina, and we had to adapt a lot because they change a lot depending on where they are. So like we would be, I believe it would be more than easy to provide it to you. But yeah, I believe that you would have to induce spawning in the lab. And that's the thing, like you cannot just rip the gonads and fertilize them in vitro. It's better once they spawn because they fertilize better. And even though they have like a real high mortality rate, especially up until the veliger stage, usually after that, like they start to die less. So that's also a little bit of a bottleneck. But once you go through all that, you can actually have a more consistent spawning process. You can actually obtain those veligers, and I believe you would be able to send it to her, and if the dogs are capable to detect it, it would be good. But like the veligers are real tiny. They are almost transparent, you know?
Mm-hmm.
So I don't know how much tissue is there to be detected, but obviously a dog is way better than us, so it's possible.
Yeah. The dogs are trained already with the quagga and the zebra veligers as well as the adults, and it's a higher risk of actually the veligers contaminating a water body than an adult mussel, than say dropping off a watercraft in contaminating a lake. So it's really the veligers. Yeah.
But yeah, I hope it answered your question. But like, we don't have anything like that in here, so I that's all that I can provide. Like about spawning, it's a pain in the ass, but once you get the hang of it, it goes quite smoothly.
There you go. Yeah, thank you, Yasmin.
You're welcome.
One person asked, do you have any data about spread via migrating birds? Are they able to latch onto birds?
No.
Okay. All right, thank you. So for all of our guests out there, I want to make an announcement. The California Department of Fish and Wildlife is having a invasive mussel agency summit happening next week, January 28th from 9 to 12:15. This is for water managers. And I will post some information in the chat. You can view the agenda and you can register for it. But I think this is a very appropriate, presentation or event to attend if you are a water manager. So anyways, back to the discussion. Let's see if there's any questions. Okay, no questions. I think I might have a few. Okay, so as I understand, the most of the funding that you're getting are from the energy companies. Is that correct?
Yes.
Have there been any other, and you also mentioned that there's a crowdfunding portion as well. Like, what are some of the other sources of funding that your company is trying to get?
This was like, it was before we started to develop this technology, the crowdfunding. It was during the PhD of one of our team. So the crowdfunding was done to do the first version of the genome of the golden mussel. And it was like, Jesus, it was more than 10 years ago. And then later when we start to propose this technology, we search these companies, because here in Brazil you have a law that is mandatory. These companies, they have to invest in research and development, 1%, if I'm not mistaken, of their profits. So they are always open to finance, but usually they are more open to finance projects regarding engineer and things more related to their operation, but they also support environmental projects, especially for golden mussels, since it's a huge problem for them, as Mauro showed earlier.
Okay. All right, thank you. Lee asks, is there any data regarding water depths and population densities? Are they known to occupy certain areas of the water? Do they prefer certain areas?
Yes, I believe there are. I just don't know them by heart, but we can maybe mail papers if you are interested in get this data. It's not our data, but I think we have been read about it before. Yes.
Okay, thank you. Hans, do you have a question?
Yeah, I have a question again about the gene drive. So my understanding then from what you said is where you are is you have, you created a construct that uses a GFP as a reporter gene, right? So that you can see whether or not proof of concept, or are you using GFP in conjunction with your gene drive, your allele that
the second one. So GFP is part of the construct that contains all the other elements which are required for the gene drive work.
Okay, so that allows you to see the GFP allows you to see right away whether or not it's worked. Is that right?
Yes, it's to track.
Okay, and you said that you're only in the veliger stage, so right now you know that veligers are expressing the construct. Is that right?
Yes, but we also try, since you're yet not able to grow the veliger, the modified veliger until the adult, we also try to modify the adult. So inject the construct in the gonads of the adults, and we check that we are able to see also the expression of the construct in the gonad cells. Okay, so we have both results.
transfecting the embryo and transfecting the adult.
All right. And so the next step then is trying to get the villagers to survive and then eventually reproduce themselves.
Yes, yes. And also to see, because we made how to say transient transfection, so we also need to be able to make a permanent modification in the genome. Okay, so
Okay.
this is the next step.
All right. Good. Thanks. Thanks for the clarification.
Sure.
Thank you for the question. All right. Well, I think this is just about starting to wrap up. If there's any final thoughts or questions, let's hear it.
Thank you very much for your time today.
Thank you.
Yes, thank you to all the staff from BioBureau who joined us. Really appreciate you spending extra time for us.
Thank you guys for the interest.
Yes.
Thank you.
And thank you for having
Bye-bye.
us.
All right, well, that concludes our session. Please keep an eye on your inbox. We will send out the recording as soon as we can, so you can get caught up on what we missed. So thanks so much for everyone attending, and we'll be in touch. Thank you. Bye.
Bye-bye.